Hello and welcome to my channel. Today in this video we are going to study about glycogenesis. This is an overlapping term, glycogenesis. That means glycogen formation from glucose. Glycogenesis process occurs during fed state, thus leading to high blood glucose level. Glycogenesis is one of the pathway of glycogen metabolism. We have already discussed glycogen catabolism, that is glycogenolysis, in our previous video. I will put the link of that video in the description box. Since during fed state when carbohydrates convert to glucose, so the high blood glucose level causes insulin hormone to be secreted by the cell. This insulin helps regulate glycogen metabolism. It takes place in two organs, liver and skeletal muscle. The amount of glycogen that can be stored in liver is around 100 grams, and the amount of glycogen that can be stored in skeletal muscle is around 3 to 400 grams. Now, once the levels of glucose are high outside the cell, with the help of GLUT2 transporters in liver and GLUT4 transporter in muscles, glucose can enter inside the cell. Once inside the cell, glucose molecules form bond with each other in the glycogen metabolism process. The two types of bonds are formed. We'll be discussing them before we start the glycogen anabolism process. This is glucose molecule with carbon number 1, 2, 3, 4, 5 and 6. And this is another glucose molecule with carbon number 1, 2, 3, 4, 5 and 6. The bond which is formed between these two molecules is called alpha-1,4 glycosidic bond, because carbon number 1 combines with carbon number 4. Both the OH are on the same side. That's why we call this as alpha-1,4 glycosidic bond. If this OH would have been in the opposite direction, we would have called it beta-1,4 glycosidic bond. Now there is one more bond which is formed between two glucose molecules, that is called alpha-1,6 glycosidic bond. These bonds help in the branching of glycogen molecule. So carbon number 1 of one glucose combines with carbon number 6 of another glucose to form alpha-1,6 glycosidic bond. Once glucose molecule enters inside the liver cell with the help of GLUT2 transporters, there is an enzyme over here called glucokinase, which helps in conversion of glucose to glucose 6-phosphate. Glucokinase is a very specific enzyme and it works only for glucose. On the other hand, one alternative enzyme called hexokinase, which is present in almost all the cells of the body, which help in conversion of glucose to glucose 6-phosphate. And that is a non-specific enzyme. It is meant for all hexoses. This glucose 6-phosphate can convert to glucose 1-phosphate with the help of the enzyme phosphoglucomutase. Glucose 1-phosphate then combines with UTP, that is uridine triphosphate, a nucleotide. And with the help of the enzyme UDP glucose pyrophosphatase, UDP glucose is formed. This UDP glucose, with the help of the enzyme glycogen synthase, combines with the glycogenin protein. Glycogenin, being a protein molecule, has an amino acid called tyrosine, which has a OH group in it, and that OH group has affinity for glucose, due to which glucose molecule combines with glycogenin. The bond between glycogenin and first glucose molecule is not a glycosidic bond, whereas the following bonds are called glycosidic bonds. The linear structure of glycogen is formed with the help of the enzyme glycogen synthase, because it forms only alpha-1,4 glycosyl unit, whereas the branching enzyme called alpha-1,6 glucosyl transferase helps in formation of alpha-1,6 glycosidic bond. Glycogen molecule is a highly branched molecule containing more branch structure, that is more alpha-1,6 bonds, and thus the structure of glycogen can be stored in less space in the liver and skeletal muscle. That's all for the video. Thanks for watching.